Welcome to this GCSE Biology revision video on photosynthesis. We'll cover everything from the basic equation to limiting factors and how plants use glucose. Here's our revision roadmap. We will explore where photosynthesis happens, the equation, limiting factors, and the uses of glucose produced. Photosynthesis is a chemical process that occurs inside plant cells, specifically within the specialized organelle called the chloroplast. These cells are mostly found in the green parts of the plant, especially the leaves. Chloroplasts are small, green structures in plant cells. They contain the green pigment chlorophyll, which is the site of photosynthesis. Chlorophyll absorbs red and blue light, reflecting green light, which is why plants appear green. Chlorophyll is the light-absorbing pigment. It captures energy from sunlight and converts it into chemical energy, driving the photosynthesis reaction. Without chlorophyll, plants cannot photosynthesize. Chapter 1 focuses on the inputs and outputs of photosynthesis. Let's start by understanding the reactants that go into the process and the products that come out. A plant needs carbon dioxide, water, and light energy. CO2 enters through stomata, water is absorbed by roots and moved through xylem, and light energy provides the necessary power for the reaction. Carbon dioxide enters the leaf through stomata, which are tiny pores on the underside of leaves. Guard cells control their opening and closing to allow carbon dioxide in during the day and prevent water loss at night. Stomata also allow oxygen to escape. Water is absorbed by root hair cells via osmosis due to their large surface area. It then travels through the xylem vessels up to the leaves, where it is used as a reactant in the photosynthesis equation. Oxygen is a byproduct of photosynthesis and is released into the air through the stomata. This oxygen is vital for the respiration of most living organisms. Oxygen release provides evidence that photosynthesis is occurring. Now let's look at Chapter 2, the photosynthesis equation. This is one of the most important things to memorize for your GCSE exam. The word equation is carbon dioxide plus water yields glucose plus oxygen. Carbon dioxide and water are the reactants, and glucose and oxygen are the products. Light energy is required for this reaction to happen. The balanced symbol equation is 6 carbon dioxide plus 6 H2O, with light energy over the arrow, yielding C6 hydrogen oxide plus 6 O2. Higher tier students must know this. Foundation tier students must know the word equation. Light energy is the essential input that powers photosynthesis. Chlorophyll absorbs this energy to break apart water molecules and combine carbon dioxide and hydrogen oxide into glucose. Without light, photosynthesis cannot occur. Chapter 3, Limiting Factors. We will explore the factors that control how fast photosynthesis can occur, a popular topic for exam questions. A limiting factor is anything in short supply that slows the rate of a reaction. In photosynthesis, the rate is restricted by the factor at its lowest level. The 3 main limiting factors are temperature, light intensity, and carbon dioxide concentration. As light intensity increases, the rate of photosynthesis increases up to a point. More light means more energy is available for chlorophyll. At low light levels, light is the limiting factor. Eventually, another factor, like carbon dioxide, becomes limiting and the rate levels off. Temperature affects enzyme activity. At low temperatures, enzymes have less kinetic energy. The optimum temperature for photosynthesis is around 25 to 35 degrees Celsius. Above 40 degrees, enzymes denature and the rate stops altogether. CO2 is a reactant, so higher concentration means a higher rate. When carbon dioxide is scarce, the reaction slows, even if light and temperature are ideal. Greenhouse growers often increase carbon dioxide levels to boost crop yields. To summarize the limiting factors, low light slows the rate, increasing light speeds it up. Enzymes work faster as temperature rises, but denature above 40 degrees. More carbon dioxide increases the rate until another factor limits it. Welcome to Chapter 4, Uses of Glucose. Once plants produce glucose, they use it in several important, examinable ways. Glucose is a versatile molecule. Plants can use it for respiration, convert it into other molecules, or store it for later. We'll cover each of these vital processes. Use 1 is respiration. Plants use aerobic respiration to release energy from glucose, which powers growth, reproduction, and all other life processes. Photosynthesis and respiration are opposite reactions. Use 2, stored as starch. Glucose is soluble, so plants convert it to insoluble starch for storage. Starch is stored in leaves, seeds for germination, and roots or tubers for long-term storage. Use 3, making cellulose. Glucose is used to make cellulose, a complex carbohydrate that forms plant cell walls. Cellulose provides essential structural support, giving plants their shape and strength. Use 4. Making proteins. Plants combine glucose with nitrate ions, absorbed from the soil, to produce amino acids. These amino acids are the building blocks for proteins, which are vital for growth, repair, and forming enzymes. Use 5. Converted to fats and oils. Plants convert some glucose into lipids for energy-dense storage in seeds, and for waterproofing leaves as waxy cuticles. Lipids store more energy per gram than carbohydrates. Here are all 5 uses of glucose at a glance: respiration, starch storage, cellulose for cell walls, proteins using nitrates, and fats and oils for energy storage. Remember, the key concept for exams is that photosynthesis stores energy in glucose. The energy comes from sunlight and is locked in the chemical bonds of the glucose molecule. Finally, let's compare photosynthesis and respiration. These are opposite reactions. Notice how the products of one are the reactants of the other. This is a common exam link question. Now let's look at investigating the rate of photosynthesis using the pondweed practical. We use aquatic pondweed, Elodea, to measure the rate by counting oxygen bubbles produced per minute. When interpreting graphs, a rising section means the variable is the limiting factor. Aplateau means a different factor is now limiting the rate. For temperature, a sharp drop above 40 degrees Celsius indicates enzyme denaturation. This flowchart summarizes the full process, linking the inputs, structures, and outputs of photosynthesis. It is perfect for describing the entire process in a longer exam question. Here is a key vocabulary checklist. Make sure you can define terms like photosynthesis, chloroplast, chlorophyll, stomata, limiting factor, starch, xylem, and denature for your exams. Finally, here are 5 top exam tips. Know both equations, explain graph plateaus by naming the new limiting factor, link temperature to enzyme denaturation, memorize the 5 uses of glucose, and remember that photosynthesis stores light energy as chemical energy.